So, välkomna hit. Very welcome here. We have a fantastic audience here today with a school class from Varje gymnasium. Super welcome here. You are our VIP guests today. We also have an audience online. Very welcome to you. Hope you are with us from everywhere because this is such a wonderful day. So today we have also a very special guest here, Claes Wiman, and he is actually not just a special guest, he is like a really special guest because he's one of the few people in the world right now who knows what is going to happen in some minutes at the Karolinska Institute here in Stockholm. Someone will be announced to have received the Nobel Prize in Medicine or Physiology 2022. And you just come from the meeting where you took the decision, actually.
Yes.
So, can you tell us who it is?
It's very exciting, of course. It's very interesting. We will, of course, discuss later more about the prize. I think this it's very interesting, and I mean, when you participate in this process, you really learn a lot about different areas of medicine and physiology. So it's really very interesting and to be part of this.
So Alfred Nobel, so this class who is in the room today, they know a lot because they have been here for a while already and been marinated in Alfred Nobel and the Will and the prizes. But all of you out there who are listening are not maybe the same expert as these experts down here, but Alfred Nobel actually wrote already in his will that the medicine prize or medicine or physiology should be decided on the Karolinska Institute here in Stockholm, and that's where you are a professor. But can you tell us why is it called the Nobel Prize in Medicine or Physiology?
I think it's in medicine. You can think about it, health and diseases. Physiology, it's more normal processes in the body. So I think it means that it covers both the normal, how things should be, how things work, how the body works normally, and of course we know there are many diseases and there are health problems, different kinds, infections, cancer, cardiovascular disease. So it's also, I mean, over the years, Nobel Prize in Medicine or Physiology has been awarded to both basic science and you can say more applied, or where, for example, new treatments of diseases. So it's a kind of a spectrum from the very basic normal processes to more medicine or disease-focused research.
Thank you for clarifying. But I also wanted to ask you because. So Alfred Nobel, he wanted to reward someone who has made like a great benefit to humankind.
Yes.
But how do you decide then what is a great benefit to humankind, and why do you deserve a Nobel Prize?
Of course, this is a good question. I mean, I think the benefit to humankind, of course, that's an important part of Alfred Nobel's will. But it's of course a question: how do you determine that? And I think what we see is some Nobel Prizes are very focused on the basic discoveries, where we don't see maybe an immediate benefit directly now or next year. But as we have, I think, becomes clear when you look at how science develops. In many times, the basic discoveries are really eventually lead to an enormous benefit for humankind.
So that's why also I guess the Nobel Prize has been so recognized all over the world it's really for humanity. It's not only for us here in Sweden who will benefit from these discoveries. Yes. And that was actually quite controversial when the prize was in the start, that this was for researchers from all over the world and not only for being here in Sweden.
Yes, yes.
But today, this week, actually, the whole world is watching Stockholm. The Nobel Prize is getting a lot of attention in the world. And for us here in Sweden, maybe we think about Nobel mostly in the week of December, when we have the pleasure to invite all the Nobel laureates here to Stockholm for a fantastic week, when they receive the medal and the prize, and they also have the big dinner for them in their honor. But in the world, this week, October week, is the
Yes.
big week.
True.
And there are maybe people lying awake, especially if they're not in Sweden,
Yes.
where the time zone is different, wondering if someone is going to call them.
Yes, I think so. I think so.
Yeah. So was this year, was it a difficult decision, or was it like, oh, this feels like, yes, we nailed it?
I think it's always you could say difficult because there's a lot of good science, and there's a lot of work behind these awards. I don't think it's never easy. I think it's always interesting and exciting.
So you feel happy about it?
Yes, I do. I do.
So today what happened was that you woke up and then you went to the Karolinska Institute. And what happened there when you came there?
Well, we had a meeting in the Nobel Assembly at Karolinska Institute, which consists of 50 professors at the Karolinska. The Nobel Assembly awards the Nobel Prize in Medicine or Physiology. So we had a meeting and then took a decision. And you will hear soon.
Today?
Yes, today, this morning, exactly, today. So, and you will very soon, I guess, in a few minutes.
Yeah, we will soon switch over to see. And then actually after your meeting, then someone from your group went and actually called the people who got the... And they didn't know that before?
No, I think that's how it is. You get a call from from the Nobel Assembly.
Nobel is calling.
Nobel calling. Exactly. So of course I think it's something. It's a big thing, of course, for someone
I think that phone call actually changed the life for the person who gets that call forever. Because from being just I would say not a nobody, a good researcher, but to become a world-famous researcher that actually everyone wants to talk to, everyone wants to hear your opinion about everything.
More or less anything.
Exactly. Because then you enter into the category of being a very wise person when you get this stamp. So the call today changes people's lives, and that is actually someone who just got that phone call today. And I think that I don't know if now unfortunately you have to then, in some minutes, you will have to move a little bit for the audience who is in our room to see the screen on the side. But what's happening there, that is on Nobel Forum at the Karolinska Institute, where you were previously today for your meeting. Exactly. And soon the secretary and some of the colleagues will come in to announce this prize to the world. So now is the focus from the whole world. Actually, some years ago, before internet, for you it's weird that there were a time before internet, but it was actually that people came out on the stair. If it was raining, it was not so many journalists there, but they came out on the stair reading the announcement. And now we see the members coming in. So I think we tune us down and we go to the Karolinska.
Ladies and gentlemen, it's really great to welcome all of you here. It's been a few years since it was like this. So very welcome to Nobel Forum and for the announcement of this year's Nobel Prize in Physiology or Medicine. My name is Thomas Perlman. I'm the Secretary of the Nobel Assembly. I will first read the announcement in Swedish, followed by English, and we will then present the background to the prize and then open up for questions. Nobelförsamlingen vid Karolinska institutet har idag beslutat att Nobelpriset i fysiologi eller medicin år 2022 skall tilldelas Svante Pääbo för hans upptäckter rörande utdöda homininers arvsmassa om människans evolution. The Nobel Assembly at Karolinska Institutet has today decided to award the 2022 Nobel Prize in Physiology or Medicine to Svante Pääbo for his discoveries concerning the genomes of extinct hominins and human evolution. Here he is. the laureate, Svante Pääbo, was born here in Stockholm, in Sweden, in 1955. He performed his prize-winning studies at the University of Munich in Germany and at the Max Planck Institute for Evolutionary Anthropology in Leipzig in Germany. So now I turn to Anna Wedell, a member of the Nobel Committee, who will now describe the discovery. So please, Anna.
Thank you. My glasses. This year's Nobel Prize concerns human evolution. Humanity has always been intrigued by its origins. Where do we come from? And how are we related to those who came before us? What makes us different from hominins that went extinct? Before Pääbo's discoveries, knowledge was derived mainly from paleontological and archaeological findings. Bones and artifacts from extinct relatives were found at multiple sites around the world. One famous example is the Neanderthal, discovered in 1856. Many questions, however, could not be answered by analysis of ancient bones and artifacts alone. Questions such as how we and our extinct relatives are related to each other, and, importantly, what makes us different. DNA technology offered a new way of addressing these intriguing questions. DNA holds the code of life. Gradual changes to this code, mutations, form the basis of evolution, and these mutations remain in our DNA as traces from our ancestors. Our cells contain DNA in two different compartments. The long nuclear DNA carries the overwhelming majority of our genes, but the small mitochondrial DNA is present in thousands of more copies and therefore easier to study. Svante Pääbo was from an early age fascinated by ancient human history. While using DNA technology for his doctoral studies in immunology, he also attempted, as a side project, to use these modern tools for studies of ancient human remains. He soon realized that this is extremely challenging since ancient DNA is damaged by chemical modifications. It is also fragmented and present in extremely low amounts together with contamination from microorganisms and present-day humans. Svante Pääbo therefore embarked on a long series of rigorous technological developments that lasted over 3 decades. He performed much of this work on extinct animal species, but his goal was a seemingly impossible task. He aimed for our closest relative, the Neanderthal, that went extinct around 30,000 years ago. After setting new experimental standards in this challenging area, Pääbo first set out to retrieve and analyze Neanderthal mitochondrial DNA. He obtained a bone from the Neanderthal type specimen that had been discovered in 1856. From a small piece of this bone, he managed to extract and sequence mitochondrial DNA. He compared his new sequence to present-day humans and to chimpanzees and found that the new sequence was different from both of them. This was an important milestone. Pääbo had proved that it is possible to retrieve and sequence DNA from 40,000-year-old bones. Mitochondrial DNA, however, contains very limited information, and Pääbo's ultimate goal was to sequence the 3 billion base pair Neanderthal nuclear genome, which would allow in-depth analysis and comparisons across multiple genomic regions. The human reference genome had been sequenced in a huge international collaborative effort, and the chimpanzee reference genome sequence was also available. Sequencing the nuclear genome of an extinct hominin, however, seemed impossible. Pääbo embarked on his bold plan, and for this he needed more bone. Thanks to his strong international networks, he managed to obtain 3 bone specimens from Vindia cave in Croatia together with additional material from Germany, Russia, and Spain. All along, he refined and optimized his experimental procedures. By skillful use of new high-throughput sequencing technologies and by bringing together a large international group of collaborators for advanced computational analysis, Pääbo finally achieved the impossible, sequencing and assembly of the Neanderthal genome. For the first time, important questions on our relationship with the Neanderthal could be addressed. The time of divergence could now be determined to around 800,000 years ago. Pääbo and his team also surprisingly found that gene flow had occurred from Neanderthals to Homo sapiens, demonstrating that they had children together during periods of coexistence. Importantly, comparisons could now also be made to search for differences between Neanderthals and Homo sapiens, differences that may underlie features that are unique to all humans living today. Pääbo's team also retrieved DNA from a minute finger bone found in Denisova cave in the Altai Mountains in southern Siberia. Again, they started by analyzing mitochondrial DNA, and to their surprise, the sequence was different from both present-day humans and from Neanderthals. Pääbo had discovered a completely new hominin entirely by analyzing and comparing genome sequences. This was a sensational discovery. The new hominin was given the name Denisova. Pääbo's team next analyzed the nuclear Denisova genome sequence, enabling important investigations. Neanderthals and Denisovans were shown to be sister groups, diverging from each other around 600,000 years ago. They further found evidence of gene flow from Denisova into Melanesian Homo sapiens, indicating that mixing had occurred. Svante Pääbo's groundbreaking discoveries have provided important new knowledge regarding our evolutionary history. Neanderthals and Denisovans lived in different parts of Europe and Asia for hundreds of thousands of years and adapted to these environments, while Homo sapiens evolved and lived in Africa. By mixing with them after migrating out of Africa, Homo sapiens picked up sequences that improved their chances to survive in their new environments. We now know that present-day humans in Europe and Asia can carry 1 to 2% of Neanderthal sequences, and that Asian and Southeast Asian populations carry an additional up to 6% Denisova sequences. Examples include a gene variant that improves living at high altitude in Tibetans, and sequences that affect how our immune system responds to different infections. No extinct hominin DNA has yet been sequenced from Africa, since conditions for preserving archaic DNA are poor in tropical climates. Thanks to new advanced computational methods, it has still been possible to find indications of gene flow from as yet unknown archaic hominins into Homo sapiens on the African continent. Through his persistent and innovative work, this year's Nobel laureate established a new scientific discipline, Paleo Genomics. This area is now expanding dramatically. After the discoveries cited here, Pääbo went on to generate genome sequences from many additional Neanderthal and Denisova individuals. In parallel, genome sequences from ancient Homo sapiens have also accumulated from different time points and different geographic locations by research groups around the world. By using Svante Pääbo's archaic genome sequences for comparative studies, important lessons are being learned regarding our evolutionary history, including how we spread across our planet. Svante Pääbo's groundbreaking discoveries allow us to address one of the most fundamental questions of all: what makes us unique? Like us, Neanderthals had big brains, They lived in groups and they used tools, but these changed very little during hundreds of thousands of years until the Neanderthals, for unclear reasons, disappeared. Homo sapiens, on the other hand, rapidly developed complex cultures, figurative art, and advanced innovations. They crossed open waters and they spread to all parts of our planet. The basis for this dramatic development must lie in genetic changes that occurred after we separated from Neanderthals and Denisovans not so long ago. By providing genome sequences from our closest extinct relatives, Svante Pääbo has provided a novel, much more recent reference point for studies of who we are and where we came from. Thanks to his seminal discoveries, genetic variants that are shared among all now living humans, but absent in Neanderthals and Denisovans, have been identified. The list of such genetic variants is not very long. Understanding their functional implications is a focus of intense research by Svante Pääbo himself, but also by other scientists. Thank you.
Wow. I have these goosebumps all over. I just said to Klaus here that what a prize for the origin of us as human species. So I don't think you need the question of why is this awarded the Nobel Prize, but I wanted to ask you, why is this awarded the Nobel Prize in medicine?
I mean, it's about human evolution, so it's about us humans and how we are related to an extinct hominin, or two extinct hominins. So of course it has to do with our how we have developed our genes, and that of course also is very much related to our how we I mean medicine and physiology. I mean the normal function of our bodies and diseases eventually. We don't know this yet, but this comparing with the genomes of these other hominins may tell us something important, or will tell us something very important about ourselves and as humans.
So he actually extracted really old DNA, like really old.
Yes.
And then developed to look where it came from.
Yes. I think we should understand that most people thought this was completely impossible to do. So he had this idea that there should be some small amounts of DNA left in, let's say, very ancient bones, 40,000-year-old bones from a Neanderthal. Nobody thought it would be possible, but he thought, no, it might work, or it should work. And of course, the problem is really technically very demanding, because you have to get very small amount of DNA, and we should remember DNA is degraded and modified over such long times, of course. And so our genome is 3 billion base pairs. Okay, it's very big, but here, in these old bones, you would get fragmented DNA, very short fragments. So all of that had to be sequenced and then put together to a whole sequence, which is a really magnificent achievement, I think.
Yeah, really, it's like a huge Christmas puzzle, but like huge.
Yes, enormous.
Enormous. So I will turn to our VIP guest here, who has prepared a lot of questions for you. You should really take the advantage of this, because now you have an expert here, so just come out with all your questions. I also want to say to you who are with us online that we have possibilities to get questions also from you. So there is a code on the menti.com that I think is visible for you who is watching online, right? So I don't need to say the code, right? Because you see it. Great. So now I turn to our wonderful guests here. So who can pop the first questions here? Perfect. We have a microphone. Please say your name and...
Hej, Jennifer heter jag. Jag undrar vad det här kommer innebära för framtiden.
Yes, that's of course a very good question. I mean, first of all, it helps to understand who we are in relationship to the other hominins. And of course, the interesting question is, how are we different? And I think we already know, as mentioned by Anna Videll, we already have, there are already very specific variations identified. Not very many, but there are some. And in the genomes, differences between our genome and the Neanderthal genome. So we still don't fully understand what this means functionally, but I think this is ongoing research. It will be very interesting to see, to learn more and understand more how we are different and how that affects the way our bodies work and our brains work and so on.
So understanding the past also help us to understand the future.
Definitely, yep.
Perfect. So we have another question here.
Okay, my name is Anton, and I'm wondering if it was an obvious decision for the committee, or was it hard to?
Yeah, I mean it's a very good prize, and of course, but there are also it's a lot of work behind a Nobel Prize. Of course, a lot of work by the Nobel Committee to select the candidate. I think it's never easy. It's always a big effort. It's very careful. So of course you could say there are many candidates each year, and so who should really get the prize? This is a big task to make that selection.
And of course, the Nobel Prize can only be awarded to X amount of individuals, even if he has collaborated with many groups.
Yes, yes.
Of course.
So it's up maximum 3 persons can get the Nobel Prize in 1 year, and so it can sometimes be huge collaborations. It's sometimes hard to know who has done what. For this year's prize, it's very clear that the laureate has been central for the whole. For this work.
Thank you.
Do you have any favorite, commonly unknown facts about this Nobel Prize winner or any other?
Unknown facts? Not really. I think the basics, the most important aspects were really well presented by Anna Videll. I think we will see, there will be new things coming out of this that we are just beginning to see now. I think that's the most exciting to me. So in the next 5 years or so, maybe next 10 years, we will see something really interesting coming out, especially with helps to clarify how we are different from the other hominins.
But he has a famous father as well, right?
Yes, I think he should tell more about that.
Yeah, I think he's related to another Nobel laureate as well. That could be an unknown fact.
That's right.
Yeah. So we have a first here and then we come back.
Ja, jag heter Elsa och jag undrar vad det här har för betydelse för forskning i Sverige eftersom att han är från Sverige.
Svenska eller respond?
Yeah, I think we take it in English since we have an international audience, but it's fine to ask the questions in Sweden.
So the significance for research in Sweden, the laureate is from Sweden. He's now working in Germany, and I don't think it makes such a really big difference because science is international, so you can have collaborations even though you work in Germany. You can collaborate with people in Sweden, or it doesn't really matter which country you're in. Maybe he has, because of his ties with Sweden, he may visit Sweden often, I don't know. But I really think it's very exciting potential for collaborations and further work to move this, to see, and we'll see how this field develops. I think it will be very exciting.
Thank you. And we have...
How big do you think this field of paleogenetics will become because of the limited resources? I mean, genomes are not reproduced all the time. It's kind of hard to find them.
Sorry, how big the field?
Yeah, like the general research field. I think it was kind of hard for him, even though he had all these connections to get all the genomic material to Leipzig.
Yes.
So it's not like there's unlimited resources to proceed in his research.
Right. I think. I mean, if the field is considered interesting, and if it generates interesting information, I think there will be funding in one way or the other. I think the laureate Ante Päbe has really opened a new field, as was mentioned here in the presentation, Paleo Genomics. So we already see that, I mean, not necessarily other hominins, but ancient human, Homo sapiens DNA from bones and so on, can give a lot of information, how people migrated. I mean, as here in Sweden, who came to Sweden, who migrated to our country, that is now our country, so we can understand many things in the past about our own history. So it's already I think a field that's growing, a very interesting field, growing rapidly, thanks to Svante Pääbo.
Great. And we have another question.
Could you elaborate on why this discovery is so important to us?
Yeah, I think as we have already been discussing a bit, it helps us to define who we are, because now we have a genomic sequence. Of course, the human sequence was already known. That was determined around the end of 1990s. Now we have a full genomic sequence of Neanderthals and Denisovans. So we can see who we are. I mean, how are we different, basically? And then, as we discussed also, we can try to understand what these differences mean functionally, affecting how our bodies work, and for example, the brain. And we can understand why, if you compare, Neanderthals are very similar to us. They have big brains. They could use tools, and they existed over many hundreds of years in Europe, but they did not spread across open seas, to spread all over the planet. They didn't show the same, there are clearly differences between us and them. So to understand why we developed in the way we did, whereas they did not, I think that's really interesting.
Great questions you have. So we have another one here.
Hello, I'm Isaac. I'm wondering if you could use this technology to bring back extinct species from prehistoric eras.
That's an interesting question. Of course, you need the development here, which is awarded, is the characterization and sequencing of ancient DNA. But again, what you get from ancient bones is usually very fragmented DNA. Now to create and revitalize species is much more complicated. So I don't think it's in the near future, but I know people are discussing this and attempting to. So we'll see. You don't think we will see Neanderthals starting walking the streets? No, I think it doesn't seem realistic, but... We have another question.
Have you heard from him today? Have you called him to save one? Or does he know he have one?
He has been contacted. Yes. Not by me, but I think Thomas is the one who talks. Perlman, yes. He's the one who gets that happy job to call the laureate if they find them, because sometimes they actually don't find them, and then they need to go and make this announcement, and the laureates they find out by some friends or family or the press who calls. So we will hear what the story was for his call when he got his life changed. So do we have any? We have another question. You're great with questions.
Hey, I'm Midian, and I'd like to know if you think that this connection with the Neanderthals can help us to understand how we can survive better now that the world is changing a lot, and understand how they get extinct will help us to figure out how to deal with all these climate changes and everything, and be better humans.
Well, that's a very good question. I think, again, we think we will. The information or this research will tell us more about how we are different from Neanderthals. I don't know, we'll see how that can help us. As of course the world is, we have a changing climate, we have huge problems, of course, in the world today. So we'll see. It's not easy to say for sure right now how this research will, be helpful for these big problems that we have. I think that's something that lies in the future. Yeah.
Mm-hmm. Thank you. And we have a question, I think, over here.
Yeah. Hi, my name is Meike. I was wondering, is this technique used on animals as well, or do you think it will be used on animals, like other than humans?
Indeed, yes, it has been used to study extinct animals as well. There was actually Svante Pääbo got his training with Alan Wilson, who was the pioneer in this studying, ancient DNA, and it has been done. Mammoth, for example. Definitely, I think there are already sequences available from several extinct species. Mm-hmm. So it's in theory, or basically it's the same thing. DNA is DNA. So if you extract from a human bone or an animal bone, technically it's not a big difference.
But also, wouldn't you say it's quite rare that one researcher starts with a question, then find the method to answer the question, then answering the question, and then putting the pieces together, like the whole chain?
That is very impressive, exactly. I think you need to have a very strong vision of something, and you really have to work hard for it. And I think that's what Svante Pääbo did. He had this vision of characterizing DNA from extinct humans. Most people thought it was completely impossible. But he did not give up. He continued. He pursued this idea. And I think very importantly, he was able to improve technique. I mean, this was a technological achievement, really amazing technological achievement, to be able to extract this very ancient and fragmented DNA and then establish the complete sequence. Remember also, it's very easy to get contamination. So you can get some DNA from an old bone, let's say, but it's very likely that there will be contamination.
By some frogs or
bacteria, or of course, what he also established early on was that when you take such samples, you need to amplify the DNA by PCR, and it's very easy if you don't have very strict routines, you can so easily get contamination, and then you end up with human DNA. Of course, that's not going to help you. That's going to confuse your results completely. So, and bacterial DNA and so on. So this contamination was a big problem, and I think he really solved that and established very strict methodology for this type of analysis.
And that is also something that we hear from many Nobel laureates, that they had a question or an idea, and they wanted to get the answers, and no one believed it, and they said, you should give up, this is no use, go and do something else. Yes. Even the experts in the field saying that. But they always continue, and they want to have the answer. So that is really to you. If you have something that you really want to do, and everyone around you say no, and you really believe it, you should go for that.
Don't give up.
Exactly, don't give up, and then you will be there or somewhere else, but follow that, and that is actually what many of the laureates says, follow your passion.
It's not easy, of course, because you will have trouble, and you will have to convince other people that it will work, and people will not believe you. So then you have to get, and maybe it's hard in the research world, it might be hard to get funding. So you need to have some kind of positive results to get more funding and to be able to continue. So it's a struggle.
It's a struggle, but I think we are just checking if we have time for one more question, and then I will also turn and see if we have some online questions. But I think we have one more question here.
Yeah, my name is Victor Berg. I wonder if, even though people are told to give up at times, did they have a lot of support in his work?
That's a good question, of course.
Yes, yeah. I mean, you may be. I'm sure that happens a lot. People are told to give up, but it's also I think in the research world, what's very helpful if you have a mentor or somebody who can who believes in you, somebody who is who can help you and give you emotional, moral support. It's always good.
And as you said, you always need to find some funding for your research, so you need to convince someone to also fund you.
Yeah.
But we heard many stories about people who sort of gave up on funding. They went home and built things in their living rooms and other things and just continued. So you have to be a little bit maybe going outside the comfort zone to do this. I will check with the online questions here.
Yeah, we have a lot of online questions, and I will start with this one. If you know very well how human DNA has developed until today, is it possible to say something about how our DNA will change in the future?
This is an interesting question. I think we can tell that there is a certain rate of mutations in DNA, but we don't know. I mean, the changes in terms of mutations, genetic changes, occur in a random fashion, more or less. So we cannot really predict how evolution will develop. We only know that, I mean, evolution, what it's about is that you have a variation you have a genetic variation, and then you have a selection imposed on that. So, but it's not possible, I think, to really make strong predictions on which direction that will go. Directions.
Thank you.
All right, I have another one here. Can this research be used for developing medicines in some way?
Yeah, that's interesting. We cannot say that now. I think it's very hard to predict that right now. But again, I think when we study these differences between us and other hominins, can tell us more about how our bodies and brains work, and this could perhaps lead to important treatments in the future. But of course, this work is not really focused on diseases. It's focused on physiology, or really the genomes, the genetics of humans and human evolution. So it's a bit far from diseases and new therapy right now, at least.
Thank you. So another one. Why do they analyze mitochondrial DNA? What is the difference between mitochondrial DNA and ordinary human DNA?
That's a good question.
So maybe just saying, what is mitochondrial DNA compared to human DNA?
Yes. So of course, in a cell in our body, we have a nucleus. That's where most of the DNA. That's a nuclear DNA, which is 3 billion base pairs. That's where essentially all of the DNA is. But we also have mitochondria. Not one, but we have maybe thousands of mitochondria in each cell. These are the sort of factories that generate energy in the cell. There's some DNA in mitochondria, so there's a small genome also in mitochondria, much smaller, only around 16,000 base pairs. So it's a tiny fraction of all DNA. But since it's present in many copies, it's easier, and it's small. I mean, it's short. So that's why it was a good choice to start out with mitochondrial DNA, because it's much easier to get sufficient amounts. But it was, of course, just a start. So then, of course, Svante Pääbo wanted to move on to the nuclear whole genome.
Of course, in the beginning, he just found really tiny amounts of DNA, and then, of course, he had larger amounts to amplify and to study for the mitochondria.
So when the technology developed, he could move on to the nuclear DNA.
So we have one more question,
we can have this one as the last one. Was this the first nomination for Mr. Pääbo?
I don't know if are you allowed to talk about this?
I cannot really discuss that, I think.
Because then otherwise we need to, then you can't get out of here if you know something.
I know.
I think it's actually the work behind the prizes are secret in 50 years. And after 50 years, and since Svante Pääbo is not that old, I think he was not nominated before 50 years back. So
50 years ago, he was not so old.
No, he was not so old. So I think that if you come back, like in 50 years, we can discuss that. So I think this has been a wonderful day so far. I'm like super excited for this award, actually, because it's such a great researcher, it's such a great discovery. the whole work that he has been doing, from his questions to developing a technique to answering to a completely new field, to give us an idea of where we come from. That is like, that is what science is all about, actually. So I'm super excited today. And I would also want to say that everyone who is watching, this week is like it just continues in this like wonderful feeling, because tomorrow we have a new prize, and on Wednesday we have a new prize. We have new prizes every day this week, and on Monday we have the last one in economical science. But tomorrow you are welcome back online, and also to the museum if you want to visit us. And it starts at 11:45 tomorrow, and it's in physics tomorrow. So we look very much forward to that as well. But first, I would want to say a huge thank you to you that you came here and shared all your expertise and knowledge and excitement about this year's award. And especially to you, of course, our VIPs today. You had such good questions. I'm like getting this super excited by having you as our future taking care of things or investigating or doing whatever you want. But follow your passion and big things will for sure come out. So, yeah, I think we just end by here. So thank you so much for being here with us. Thank you. Thank you.
Thank you. Great pleasure.